Thank you, Dr. Woodruff, for the opportunity to take part in this panel discussion as the leader. It gave me the opportunity to really think deeply about where we are after this 1-year mark. So welcome to this panel discussion. We'll be talking about really incorporating sex into the research agenda. So going from policy to now the implementation phase. So we're joined today by 4 leaders in this area. Dr. Larry Cahill is a professor in the Department of Neurobiology and Behavior at the University of California, Irvine. Dr. Bethanee Schlosser is an associate professor of dermatology and obstetrics and gynecology, as well as the director of the Women's Skin Health Program, Vulvar-Mucosal Specialty Clinic, and Skin Concerns and Pregnancy Clinic within the Department of Dermatology at Northwestern. As you heard from Dr. Woodruff, she is the founder and director of the Women's Health Research Institute. She's also the vice chair for research and the Thomas J. Watkins Professor in OBGYN at Northwestern. And finally, Dr. Catherine Woolley is the William Deering Chair in Biological Sciences and a professor in the Department of Neurobiology at Northwestern. So this is truly an exciting landmark now that we've had 1 year of this policy. And so now we're entering really the heart of implementation. So I'm eager to hear from our panelists today about their thoughts on some of these high-level thought questions that come after having this year to be present with this policy, but also to consider some practical points that might help researchers in implementing this policy in their own research. So I have a series of questions which I'm really excited about, but I also want to make this a dynamic discussion. So please feel free at any point in this discussion to come to the microphone, and I will be sure to make sure that your questions are addressed or your comments and feedback. So we're going to start with some questions, and we're going to go from more of the conceptual to the practical. So Dr. Janine Clayton, who is the NIH Associate Director for Research on Women's Health and Director of the NIH Office of Research on Women's Health, has mentioned that considering sex as a biological variable is not the same as looking for sex differences. So I would like to ask the panelists, in this new policy, really what does considering or accounting for sex as a biological variable mean? So I know Dr. Woodruff listed some of what this consideration means, but there seems to be a very large variability in the range of these suggestions. And to put it more simply, what advice would you give investigators who are just starting to adhere to this inclusion policy? Do you have any takers?
I can start by saying a few words.
I should mention, sorry, could you start, I forgot to mention with the introduction, could you just explain a little bit of background about how sex inclusion is important in your research or your clinical practice?
Okay, I'll do that. So I'm Catherine Woolley. I'm a basic neuroscientist. I study synaptic plasticity, which is sort of a broad term that refers to changes in the strength of connections between neurons that underlies learning and memory, and that when it goes wrong can lead to a variety of neurological or neuropsychiatric disorders. I think I'm here and included in this group today as a representative of the relatively recently converted. Like many scientists, including neuroscientists, I spent most of my career studying only one sex. In my case, it was females, not males. And I really considered that to be fine until we began to develop results in the lab on experiments on the brains of rats and mice, where the results did not match with very similar experiments that were published already in the literature. And it took me not too long, but a little while, to consider the possibility that the reason our results didn't match with almost identical experiments that had been published by multiple other groups was that we were using female animals. And most likely, and in most cases, though not explicitly stated, those other experiments were done using male animals. And so we tested that hypothesis by using male animals, and we got identical results to those that were published in the literature and presumed to be true generally for both sexes. And so we stumbled accidentally across a series of pretty profound differences between males and females at a molecular level in the way synaptic plasticity works. And in that way, I converted myself into an advocate for considering sex as a biological variable in our research in the lab. I do think we should all try not to interpret the NIH policy to consider sex as a biological variable too narrowly. What consider means is really going to be different for different people, and I don't think there's a specific formula or rule that has to be followed. I think in the context of a grant application, one really should make a genuine effort to consider whether sex is a variable that is or is likely to be relevant to the outcomes of the experiments that are proposed. And I think it's not too difficult to take the type of approach that we take now in our lab, which is simply to begin an experiment with the presumption that there is no difference. We split the groups 50-50, males and females, and we keep track of which data come from which sex. And if the data are perfectly overlapping, no difference between males and females, we report that way. And that rationalizes continuing to mix males and females, right? We always keep the data separately, but you either demonstrate that these are two groups that statistically differ from one another, or they're not. And I think in many cases, there won't be any difference between the sexes, and then there's no difficulty in using both sexes and putting them together. And in some cases, there will be differences, and in those cases, it's incumbent upon the researchers to report that and to be clear about which results apply to which sex. And I don't really think it needs to be much, much more complicated than that. There will be times when it makes sense for a research program to delve more deeply into those differences, and many times when it won't. And so that's the approach that we take in my lab.
I could follow up on that. Is this loud and clear? It's not sure. Can you hear me? This is on. I'm Larry Cahill, and by way of background, I too came to this issue through my own research. That's how most scientists get convinced of something. 15 years ago, I had done what all my neuroscience colleagues do. I had studied only the male my whole career, thinking that was a perfectly reasonable thing to do. And I had shifted from studying rats to studying humans, and taking the theories that we thought we had some understanding of in rats and extending them to humans. And when I did that, I started to study this thing called the female for the first time in my career. That led to some discoveries about sex differences, which I'll mention in my talk. And then I came to a choice point around the year 2000. Explain these differences I was finding or explain them away. And I was encouraged greatly to explain them away, but I decided I would try to explain them. Anyways, that led to my discovering what was already blatantly clear at the time and is even more clear now, which is that there's sex influences everywhere, all over the place, and this from a field that's overwhelmingly trying to ignore the issue. So that's what made a convert out of me. I'm a little older convert than Catherine here. But, and I'm thrilled by the way the issue is starting to take root, and irreversibly so. In terms of how best to go about it, my advice for almost all the neuroscientists out there and scientists in general who've only studied males is order some female animals. That's it. And start studying them. That's how it all begins. Because you got to understand, that's not happening. That hasn't been happening for my whole career. In terms of what assumptions to begin with, here I think I would differ a little bit from Catherine. She said she has this reasonable starting point where she says, well, there probably won't be a sex difference, but let's find out. I would say something different. I would say there may or may not be a sex difference. We don't know. They have proven to be so ubiquitous, popping up in so many surprising places that you flat out don't know, down to the cellular level, down to neurons in petri dishes, whether or not you're going to find a sex difference. You don't know. So I would say you don't get to assume one way or the other that there will or will not be a sex difference. You don't know. So find out. There may not be, in which case, excellent. And in many cases, there aren't going to be any. Excellent. Let's find that out. But at the same time, we are going to discover, as you pointed out, many instances where there are, often to our immense surprise. And those are the ones we need to know about. So I think the only thing I'd say different is start out with the assumption that you don't know whether or not what you are determining in this sex will apply equally, if at all, to the other sex. You don't know. And you're obligated, since both men, we're supposed to be doing science that equally benefits men and women, you're obligated to find out. And then just, start doing the experiments and see what falls out.
I'm Bethanee Schlosser, and I'm predominantly a clinician, and I do translational work as well in the Department of Dermatology. I also am biased towards female. By virtue, I run the Women's Skin Health Program, and so for me, it's been really important and essential to remember that the patients and the physiologic and pathologic processes that I look at, I need to include the male component as well. And part of that for us has really been looking at what resources are available. So here at Northwestern, we have a very active skin disease research center. It has a keratinocyte culture bank as well, an organotypic raft tissue culture service. But as Teresa alluded to, we know that the predominant source for keratinocytes, the main skin cell, is actually male foreskin. And so that probably doesn't recapitulate the finding in females or potentially across the age span either. So we've really been conscientious in terms of helping to provide additional resources that will enable investigators at the very basic level to actually address those questions in both female and male cell types, keratinocytes, fibroblasts, lipocytes, sebocytes, et cetera. In addition, one of the benefits I think that we've seen here at Northwestern is the Skin Disease Research Center, which is really just one of 7 or so that's nationally funded here in the country. In the renewal application that was successful, we stated that we wanted to go ahead and have one of our pilot grant programs specifically dedicated to an investigator that was addressing sex as a variable in their application. And that was a kudos that was commented upon by the reviewers in terms of, as a boon in terms of our research center, and has been mimicked then and replicated throughout the country. So in my practice, the questions that I raise really stem from clinical observation. And I talk with my colleagues, and then we translate that to our colleagues at the bench in terms of saying, what's the fundamental process that's going on here? And if there's a difference epidemiologically or disease severity, response to therapy, we actually go back and say, why is this happening? But in dermatology, we have very few fundamental bench resources that can address that at the cellular level, so we're trying to improve that. At the animal level, obviously, that's available, as Dr. Woolley and others have addressed.
So my own research is in the area of the ovary, and so you would think that this would be the easiest place to say, forget about it, I'm not going to study the male. And so, but because of the policy, I've actually thought more and more about male testis, about the testis biology and how we can actually study and address fundamental aspects of, particularly in the area of the loss that is associated with chemo and radiation therapy. So we now have in our lab an entire group that's working on male reproductive biology. My own conversion event took place back during the time of the original NIH policy in the early 90s. And I was at Genentech, as I mentioned, when we were doing the clinical trials on TPA, the clotbuster that still is one of the most utilized recombinant drugs and one of the first blockbuster recombinant human proteins. And there were 2 trials, the JISSI and the GUSTO trial. One was 50,000 men, and the other was 75,000 men. And so it was then that, as one of 3 female scientists, and I'm sure the males thought about it too, but I was the one that asked, and where are the women? And I was told, well, it didn't really matter. One group was American and the other was Italian. And I thought, well, they're still all men. So that was kind of the first question. And it was at the time that the NIH was being told by Congress in that time that they had to have a policy for inclusion of females in research. And that's actually a mandate rather than a suggestion. The current notice is a suggestion. It's not the mandate that the 93 inclusion was. Well, so that was kind of the first kind of awareness that I had of it when I came to Northwestern and was the basic science director for our cancer center. I became aware that we had male patients that were sperm banking prior to the first life-preserving but sterilizing cancer treatment. And so I asked, well, oh, that's awesome. I didn't even think about it. What's happening for the women? And those same women with the same hope for survival of the disease were not being offered any fertility option. And, you know, instead of saying, well, there's a biological issue here, it was more like, oh, well, they shouldn't really be thinking about that. They should really just be thinking about survival. And to me, that represented a real gap that could be addressed by some of the research we were doing. And so that led me to begin to try and adapt some of the stuff we were working in the ovary towards that utility. But also now we include males, and particularly for the prepubertal boy who has the same lack of options that the women did when we first started our program. We're now developing research programs for those young males. I think the advice for people who are starting out is, you know, if there was ever a time to start in biomedical science, now is it. It's the most exciting time because there's a whole new opportunity to do discovery research that's not ordinary. It's actually the extraordinary opportunity to open up new dimensions that you couldn't otherwise. And you're being encouraged to do that. And so I think this is an unparalleled opportunity that's going to be a short window, because eventually this will become normal science, because it is normal science. But I would really go out and urge your PI to ask questions about males and females. In fact, one of my own students came who was doing an islet study and said, hey, look, there's a difference between the male and the female glucose response. And so there's discovery out there waiting for the scholar who really is open to this topic. And those of you at Northwestern or UIC, you really are an environment where we're ready to help support you with all of the tools and reagents that we have to help you be enabled to ask those fundamental questions that's going to really accelerate the pace of your own professional career trajectory. And so that's something that I think is a real opportunity.
So I want to ask, does anyone in the audience have any questions at this point or want to share experiences? Sure, come up to the microphone. And if you can identify yourself as well, that would be great.
Hi, I'm Haley Titus. I actually am trained in neuroscience, so I appreciated all of that commentary. And I worked with genetic modified animals, and so your points really hit home, but also to publish and to see differences in data with power, you have to increase that end. So do you think it's going to be standard, also considering things like IACUC and getting your procedures and protocols approved with reduction of animal numbers, that in papers you're going to have to show that statistical data of males and females, no difference, and then validate why you grouped everything together? I guess I'm asking, do you think the scientific rigor of peer review is going to get more difficult? Mm-hmm. Do you think IACUC review is going to get more difficult? Is there going to be funding from the NIH to support male and female studies if you're increasing your N by double? So these are just things that I'm thinking about while you guys are talking.
So first, I would say that my prediction is that rather than more difficult, I might use the term more rigorous. But that's all right. But so, I mean, you're looking way ahead toward the horizon where the things that we're talking about now as good ideas become rules that are enforced by journals or governing boards within the institution about the use of animals or studying people in clinical research. And I honestly, I don't know. I don't think we're anywhere near that happening now. And my own sense is that we don't know yet whether that would be a good idea. I think we're at a phase where, how to say, the way I like to think about it is I try to persuade scientists with science. So we are doing our experiments in both sexes, and we're going to find some things that are the same and some things that are different. If more and more people become persuaded that it is in fact valuable, perhaps even necessary, to report data in both sexes, maybe it's going to be valuable, perhaps even necessary, to power those studies sufficiently to analyze statistically whether there's a difference between the sexes, then that will proceed naturally. And those regulations, which right now seem like they would be very extreme, won't be extreme. We're certainly not there yet. On the other hand, while we're in this period of time where people are exploring, I'll tell you a story about a colleague of mine who studies mouse models of disorders, and her graduate student. This is a colleague who works on mice and studies a gene that is related to Angelman syndrome. And they studied the impact of this gene, or they intended to study the impact of this gene on the functioning of the cerebellum, which is important for motor coordination and sort of timing responses in the brain. And perhaps it's because her graduate student attends my lab meetings regularly, and we were constantly talking about these discoveries of sex differences we were making. Maybe it's because I have lunch every day with this colleague. They decided to just break down their data by sex. They said, we've already collected all these data. We had no interest in sex differences at all, but because they're using these valuable animals, they use both sexes. They just split the data, males versus females, and discovered in one afternoon not only a sex difference in the basic electrophysiological properties of these cerebellar neurons, the way they were firing action potentials, a sex-specific response to mutation of this Angelman syndrome-related gene, just by looking at data they already had collected. That's a huge advance with very little additional effort required. And I think we're going to find more and more of those types of things, even without journals requiring it, NIH requiring it.
I was just going to say, I think I was going to make the comment before, this, instead of seeing it as a potential greater burden, it may be higher yield in terms of low-hanging fruit. If there are greater gaps that you recognize, and just simply changing the perspective in the way that you're asking the question or looking at the data, this may actually be very, very valuable and actually easier than you might perceive initially. Because I think oftentimes this will present us with very low-hanging fruit, that whether that demonstrates a difference on the sex basis or not, it's still important in terms of getting that knowledge disseminated and reported.
And I would add, it's a very good question, because you've given voice to some of the fears that are out there as people are now having to actually think about this, and including some completely false things like, oh no, I have to double my N, which nothing states or implies that is true in people's research. It's just a conclusion that sounds reasonable if you haven't thought about it much, and that people jump to. But the answer to your question, as Catherine said, is the more it becomes clear to editorial boards, to study sections will be the last to go, to everyone, that not only is it justifiable to do this, it is unjustifiable not to do it, right. Then that is what we're going to do. But remember, the situation we're at now is if Catherine's colleague goes and tries to publish these data, what they may well have happened to them, and I've heard this plenty of times over the last 15 years, is they may well be told, we'll publish it, but get rid of the sex difference stuff. It really doesn't matter. That's where we are now, today, right now. That's what is quite likely to happen. And the bigger the journal, the more they're likely to say, I've had it happen to me personally. Right? We'll publish it, just take out the sex difference stuff. That's where we are today. Is that justifiable? So I think instead of being afraid of things that might happen in the future from doing what the data say we ought to be doing, we should be afraid of things that are happening right now.
I think there's one last thing maybe implied in the question, and it is basic. As scientists, we're really out there trying to make our own way. There's nobody who tells us what time to come in the morning. There's nobody who says go home at night. There's nobody that gives you an attaboy when you're writing the grant all night Saturday and Sunday and barely get up to take care of the kids or whatever. You know, we really are boats on our own bottom, and that's intellectually and what we do. And partly why we do it is because we love what we do. You know, you couldn't tell me not to get up in the middle of the night with that idea and write it down and help that student to get to the next level. But implicit in that is that we are all so autonomous. It feels awkward when someone comes and says, you're not doing something right and do this. It feels like we're being corrected. And that's what this policy, I think, for some people, feels like we're being told what to do. And I think we've got to eliminate that kind of emotional response and simply ask, what is it that once we do it, we could learn? And so are there strategies that we could participate in to make sure that our journals understand what the issues are? Because after all, for the majority of the journals, we are the publishers of those journals. And so, for example, in the Endocrine Society, when we debated this topic, it was that we said, well, the most important thing is not to tell our investigators go back and do the other sex or eliminate half the sex, but to simply report the sex. And by doing that, it changed overnight. It changed everything immediately. It's like in one afternoon, you could discover something new about a syndrome. In one vote, we were able to allow everyone to now be able to repeat the studies, because even in the title, if it's a single sex, you have to say in male animals X, Y, and Z, in female animals X, Y, and Z. And so the implicit is that it's both sexes, and the subordinate is male or female. And I think that's really where journals are going to head. I don't think they're going to get it's not going to be harder. And I would say for ACUCs, again, we are the ACUC, and so I think we, requiring power analysis to know if you're going to have an effect is something we should be doing anyway. And so that variable, adding a variable of sex, is something that I think is something we should all be very comfortable doing. And those are some of the tools that are on the website to help people understand how to do that kind of work.
So I'm going to pick up on that. That's a nice segue. So the types of resources that I've heard available that you posted from the NIH, and just also what you're developing for your clinical colleagues, that's sort of at the researcher level. But the question I have for you is sort of what stakeholders do we need to educate? I know Dr. Woodruff has really thought about reproductive science in general from K to gray, so across that spectrum. But what kind of programs do we need to implement, or who do we need to be training at what levels, and where is the status of that right now?
So I think we're all in, and I know there are members of the public here, there are students here, there are faculty here, there are administrators here. There are people who have been doing this for a long time. There are people who've never even thought about it, and they saw the title "Sex Sells" and says, "Hey, that looks interesting." So I think we all have to be educated, and I think the public very much needs to be educated as well. So making sure that we speak in language so that all of us understand, because in many ways I'm lay to what's being learned in the neuroscience literature, so I am the public to Larry and Catherine's work. And so if they can't communicate it well to me, then it's not going to be communicated to the general public well. So I think we are all the stakeholders.
I'd like to make an additional point about public education, and that has to do with the temptation to overinterpret sex differences. This happens, it's on my mind because it happens commonly in neuroscience, that let's say my lab identifies a difference in the way synaptic plasticity works at a molecular level. It's very easy for someone to make the mistake of translating that directly to a difference in behavior. And since I study a part of the brain important in learning and memory, that's easily translated to cognitive function. And that is a terrific error. There's no reason to predict that a particular molecular or cellular difference in a rat or a mouse explains some stereotype about cognitive function. And so one of the things we really need to do to train ourselves as scientists and to be very disciplined about is communicating accurately what can and cannot be interpreted from studies that consider sex as a biological variable. And that's something that I think all of us in this room need to be very careful about and disciplined in doing.
I have an example that follows up on that point very beautifully. Did I see a hand up there?
Yeah.
All right, let me. let me say my piece, and then we'll go straight to you. To amplify what Catherine just said, I have a colleague at the University of Maryland, Peg McCarthy, excellent scientist, been a leader in the field of sex differences for many years. A couple years ago, published a paper showing that a certain peptide existed in much higher levels in female rats than in male rats, and she related the levels of this peptide to the amount of vocalization that occurred in the male rats versus the female rats in relationship to the mother. These were pups. So the female rats had more of this peptide than the male rats and were vocalizing more than the male rats. It showed up in the press as, see, this is why women talk more. Okay? Now, there's no one that pissed off more than Peg McCarthy, right? Peg, if she was sitting here, would be the first person to say, yes, that's annoying, and it in no way constitutes a reason not to pursue the research. What it constitutes is a reason to get out there as a scientist and say, no, that's not what it means. Here's what it might mean. But in no way, if the public takes off and runs with something, does that constitute a reason for the scientists to stop doing their job. They just have to be more careful or get out there in counteracting nonsense that appears.
Yeah, I think the scientists have a responsibility to help each audience, whether it's other scientists, whether it's journalists, or whether it's members of the public, to understand the significance of the work that they've done carefully. That's my point.
And I think, just like others journalists, et cetera, may have an axe to grind, whatever that it may be, I think it's important in the educational programming that we do, all of us, that they understand that we do not have an axe to grind. So although I run the Women's Skin Health Program, that does not mean that I feel that women have more severe disease or greater impact on their quality of life compared to males. So I think putting that out front right away and making sure that they understand, whoever the audience is, that's really important as well.
Is there a question?
Let me try without a microphone. I'm Rob Pless, and I wanted to introduce to this assembly, which I think are mostly scientists, a bit of paranoia. And I'll use an example of Dr. Woodruff's research on oncofertility. It's my understanding that she has a good program that is successful, and she has very great difficulty recruiting people into her program from other hospitals and universities, because if they supply people for her study, then they lose a patient and Northwestern gains one.
No.
Not true?
Really?
Say that into the mic.
Yeah. No, in fact,
Paranoia solved.
the paranoia solved. The beautiful thing is that every patient has a home. They either have a home at UIC or at Rush or at University of Chicago or at North Shore. The great thing about Chicago is that we are one of the densest healthcare centers with academic leadership any place in the entire world. And so I'm really proud of our oncofertility program and the colleagues we have. We call it the National Physicians Cooperative, in fact. We have a word for the fact that we have physicians all over Chicago and all over the nation who participate in taking care of the patient where they are, so that they don't have to move between centers unless there's something specialized that hospital doesn't have. So we're in oncofertility and in many other specialties, particularly in Chicago, there's a very open, embracing way that we're not worried about losing patients. We're really excited about what we can do to help patients.
Then my inside source wasn't very accurate. Let me finish with one more criticism of scientists informing the public.
Okay.
They talk too much. By that I mean scientists want to give the absolute truth, not the essence of the truth. So rather than giving 95% of the data, they give 100%. And what the press finds is all kinds of exceptions to every general rule that scientists have, so that nobody trusts scientists because everything is an exception. This is very different from speakers that come from specific industries where they give the minimum amount necessary to convince people to give them money or to believe them. Scientists want to look for truth. How foolish.
This is why we need Stephen Colbert.
If only everything could be encapsulated in 140 characters.
I'm Kirsty Danielson. I'm an assistant professor over at UIC, and I'm an epidemiologist by training. So something I want to offer us to think about is that we need to think about clinical and biological significance and statistical significance on a spectrum. So as an epidemiologist studying 10,000 people, we find something that's statistically significant, but it's this big clinically. So, and biologically, studying a few mice, you might find a big difference, but it's not statistically significant. So I think on that spectrum, we need to think about both of those aspects as we think about our studies and interpret them and publish them.
Yeah, I absolutely agree with what you're saying and how important it is to make the distinction between statistical significance and biological or clinical significance. This is something that that scientists sometimes fail to do, and we should help them to avoid that. There can be a bias to either magnify a difference, and maybe someone wants to make a splashy headline, and they are willing to overstate a statistically significant result as meaning biological significance, and it's one of the errors that I'm talking about. It's important for scientists not to make. So that's, as members of the public or the media, it's very important to be able to distinguish between those two types of uses of the word significance.
So I wanted to ask, I know all of you are proponents and have really made some important discoveries by considering sex as a variable. Are there limitations or challenges of this policy or this concept that are either perceived or real that you could talk about?
I'm not sure where to begin. Go ahead.
So Larry and I had a conversation last night about this, and I think that it's very likely that the main outcome of this policy will be to increase the rigor of the basic science that we do, and to increase the likelihood that the results of basic biomedical research apply similarly to male and female. I can think of several limitations, and one we've already discussed, which is the overinterpretation of differences that are discovered in animals or cells as meaning more than they actually do mean, without doing experiments explicitly designed to test that. And another limitation could be, I don't have any examples of this myself, but in my imagination, since we were on the theme of paranoia, I'll confess one of my paranoias, is that some people will be motivated to excuse the issue of sex as a biological variable in their work, and may too superficially consider sex, too easily dismiss the possibility of a sex difference, publish that result, and then we'll actually miss something that is biologically significant that's different between the sexes. Those are my fears are amplifying small differences into inaccurate big ones, hiding differences that exist by doing a superficial analysis. And my hope is, and I think this is true, that the majority of scientists will sincerely address this policy, and as evidence for legitimate differences between males and females and legitimate analyses showing that they are similar become routine, those issues will go away, and this policy will have improved the basic science to translational to clinical science pipeline. But those are my two fears.
With your second fear, do you feel that sort of we're asking knowledge of someone who may have only been focused on males or females to now understand a whole lot of biology, even if we're talking about scientists or clinicians, a whole new set of biology in terms of the genetics, the microbiome, just the normal different, like fundamental biological differences. So is this going to require collaboration or a new sort of skill set?
It depends on the level at which one wants to understand the biological difference. One doesn't have to understand all of the biological mechanisms for development of sex differences in order to compare males and females in an experiment. And so I don't think that level of education about how biological sex differences develop is required of every biomedical scientist. There will be occasions when a scientist discovers something and then is motivated intrinsically to learn more about it, and then they will learn those things. But I don't think that's actually required. I think it's more important to help people learn how to test for sex effects in their experiments carefully, and that's one of the things that the WHRI has really worked hard to do, and put all those tools online to help people learn to do that.
So one of my concerns is we would lose the trust of our public. Every year, $30 billion is given to NIH, and I know there are members of the public here today. And one of my friends said, well, why are we letting the public in? This is like airing our dirty laundry. We already have a problem with scientists not really being trusted in general. And if you look 20 and 30 years ago, scientists were among the most trusted individuals in our society. And so what I want to address is that we are opening up an area that if there is significant discourse rather than simply an acknowledgement, as we often do, when science is done well, people will acknowledge it and move ahead. And the fact that there is a little bit of discourse, and I think it will become moot over time, but for right now there is, that can create a level of distrust in the public that particularly now could be very difficult to countermand. So my message is that we are on a good path. The discoveries that we've made over the last 30 years have improved our health in remarkable, tangible ways. Every dollar that's spent by taxpayers in the biomedical machinery has given back $58, whatever Colin says. I'm forgetting what the exact number is. But we have paid off dividends in our own health. But where we are now is at a moment where we are at a new inflection point. When I came into science, it was molecular biology. Molecular biology was a disruptive technology that changed everything. And we're at that kind of inflection point now as well. And so about once a decade, you can count on science coming up with something new. And so I think that that $30 billion, and I hope $60 billion, if we can double the NIH budget, would really go towards improving all of our health for a lifetime.
I think, what we've talked about is we're not looking to see that there is definitely a difference, right? So that there may not be a difference, but giving equal importance to the publication of quote unquote negative data, I think that will be really important as well. So in our, organizations as reviewers, as editors, section editors, et cetera, I think that will really help in terms of, disputing some of this. We're not giving you the entire picture. So I think that's really helpful. And my biggest fear as a clinician is that exactly what they alluded to, at the cellular level, that's going to be translated then into a message that potentially can have very negative public ramifications. So let's say that a female fibroblast tends to move quicker, multiply more readily, et cetera, compared to a male. Someone then gets that and says females heal better compared to males, so then females would do better with plastic surgery. That's my fear, and I do see that potentially happening.
That's the dermatology version of my brain fear.
Right. And that's the public perception media that's more easily translated. So.
And those are the kinds of things that happen with all of our science.
Right.
These aren't issues that are specific to sex differences. To get back to your question, which I think was, what do we see as the biggest roadblocks, sources of resistance? For me, the single biggest one, and it's not going to go away anytime soon, is the enormous bias that exists in the mind of scientists, especially the older the scientist and the more they've been at this for a long time, the bias that holds that there are no sex differences to worry about outside the bikini zone. Sure, they're in the bikini zone, but you get outside the bikini zone and it's marginal, it's due to pesky hormones. That is an enormous bias, which is currently dominating a lot of study sections at NIH. I'm reminded of the painfully probably true comment by Max Planck that science advances one funeral at a time. It's an overstatement, but it has a real degree of truth in it. And I think it's going to take the younger people in this room to do it. And I have no doubt that 20 years from now, you'll be telling your students, you know what we used to do? We actually used to think that you could get away with understanding everything you need to know about a female by studying a male. And that's, I don't know if that's more pessimistic or optimistic statement. As the younger people come along and challenge these assumptions, we're going to see it move in the direction that Mother Nature thinks it should move.
You want to go up to the microphone?
Hello everyone, my name is Sahiti Pamarthi. I'm a postdoctoral fellow. We do translational research in the Hemong department. I'd like to make a case for the pesky hormone since we're talking about it. I work on prostate cancer, and my concern is that how scientists through the years have branded themselves as breast cancer and prostate cancer scientists. My PhD was in breast cancer, and this particular subtype of both these cancers are histologically similar. The way they depend on hormones, the breast in the female and the prostate in the male, is quite similar. And we specifically study the hormonal receptors, the androgen receptor in the prostate, and the progesterone and estrogen receptors in the female. The way they work, the way they go into the cells and bind to the DNA is quite similar. And through the years, researchers who work in exclusively in one of these models have learned a lot from the other model. So my concern is that why don't we rebrand, call them just hormonal sensitive cancers, include a prostate cancer cell and breast cancer cells in the same research, because it is going to be definitely economical and also broader implications. What are your thoughts on that?
Well, I am not a cancer biologist. However, I think one of the things that we do as scientists is we look for commonalities and parallels between the specifics of what we do and related fields, and you have identified one. It sounds to me like what you need to do is deliver a talk at 2 major meetings where each side is in great attendance and make an argument for that, because it makes a lot of sense. Write an editorial piece in a journal that's major in your field.
Yeah, and I think if you go even more broadly about those pesky hormones, if we look at cell culture, which is what you're talking about, there are no people who study general cell biology that include any hormones. And so there's not the testosterone that's high in the morning and falls during the day, nor is there any background estrogen or progesterone. And so what we're really studying in tissue culture is some kind of odd postmenopausal cell. I don't even know what it is. So it does not have those hormonal influences that are part of the general body. So really understanding hormonal influences on cancer onset and progression, and actually on the background of muscle cells and liver cells and all the other cells that inform the biology of what we do is something that I really am advocating for. And I think that that's going to allow us to understand something more, but it's not a mandate. It's something that I think if people could be persuaded that there's value and that maybe what they're studying isn't quite analogous to the problem that they're trying to solve, that's when the conversion event or the light bulb would go on, and they could say, well, let me see what happens if. And I think that's going to be part of the neat thing about the new generation of scholars. The opposite of the plank is one scientist is born every day. And so those that are newly born
Thank God.
are going to be born into an environment where you have an iPhone and you only talk with your PI by iChat, and you're going to study sex as a variable. I think it will just become as routine and as that. So
I'm going to ask one last question. I view this, I'm going to say that this is going to work and that the horizon is potentially not that far away. So I view this also as very much of a big data generating, potentially endeavor, as Dr. Woolley mentioned, just in one afternoon, by considering a different aspect of a data set that had already been generated, we learned something new. And this seems to happen across disciplines. So I guess my question is there value or are there efforts in place to start creating a repository or a database of what we're learning? And I think mostly I'm making an analogy to sort of the microarray repositories that we had when that technology started to be generating, where it was a requirement that people deposited that information. So are there efforts in this field to do something similar, or would it be valuable at the ground level?
I don't think there is anything. Francesca, you always come up with the best ideas, and I think that's a great one. Of course, you know, the microarray and next-gen sequencing, we don't do X and Y, so that's one of the biggest misses in all of genetics. But I think that that would be a tremendous opportunity for this field to galvanize ourselves around. And you know, we're collecting the data basically in blogs, so we could go back and get 50
and
blogs.
All the reviews that this group has done has been, and editorials and commentaries. Is
I think it's a brilliant idea, and we should really do it.